Francis Harry Compton Crick was born in 1916 in the small English village of Weston Favell, a place far removed from the scientific revolutions he would one day ignite. His childhood was marked by an insatiable curiosity, a relentless desire to understand how the world worked. He would pepper his parents with questions, dissecting everything from the mechanics of a clock to the mysteries of the stars. This early fascination with the fundamental principles of existence set the stage for his future endeavors. He conducted his first chemical experiments in a makeshift laboratory at home. His parents, Harry Crick and Annie Elizabeth Wilkins, nurtured his inquisitive mind, encouraging his scientific explorations. He attended Northampton Grammar School, where his aptitude for science became increasingly apparent. He devoured books on science and philosophy, already grappling with the big questions of life. His intellectual energy was boundless, often leading him into spirited debates with his teachers. He pursued his formal education in physics at University College London, graduating in 1937. He began his PhD research on the viscosity of water at high temperatures, a project he later described as incredibly dull. Outbreak of World War II in 1939 abruptly halted his doctoral studies. This interruption redirected his sharp intellect towards a more immediate and practical purpose. During the war, Crick applied his physics background to the development of naval mines for the British Admiralty. He worked at the Admiralty Research Laboratory in Havant, a place of intense, secretive work. His specific task was to design magnetic and acoustic mines that could effectively target German ships. This role honed his problem-solving skills under immense pressure. He worked in a team, deciphering enemy technology and creating countermeasures. The environment was a high-stakes game of scientific cat and mouse. The experience, while diverting him from pure science, was formative. It taught him the value of collaboration and focused inquiry. He learned to think in terms of practical models and testable hypotheses. After the war, a profound shift occurred in Crick's scientific ambitions. He found himself drawn to the burgeoning field of biology. This change was heavily inspired by the physicist Erwin Schrödinger's 1944 book, What is Life? Schrödinger's work posed a tantalizing question. How could the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? This question resonated deeply with Crick. He saw the boundary between the living and non-living worlds as the next great scientific frontier. He famously stated his goal was to understand the dividing line between the living and the dead. He decided to leave the familiar world of physics behind. He embarked on a new journey into the complexities of life itself. In 1947, he began this transition, a process he called his gossip test. He was most interested in what scientists were gossiping about. He retrained himself in biology with a move to Cambridge. He first worked at the Strangeways Laboratory, studying the physical properties of cytoplasm. This work provided him with a basic grounding in biological laboratory techniques. However, his ambition was to tackle the most fundamental biological puzzles of all. He wanted to understand the nature of the gene and the secret of its replication. His quest led him to the renowned Cavendish Laboratory at the University of Cambridge in 1949. The Cavendish was a world-famous hub of groundbreaking physics research. But it was also home to a small, ambitious group of scientists turning their attention to the molecules of life. This group was formerly the Medical Research Council, MRC, unit. It was led by Max Perutz, who was using X-ray crystallography to study the structure of proteins. Here, Crick joined the unit, officially to work on his PhD on protein structure. The laboratory was a chaotic but intellectually vibrant place. It was filled with spirited debates and the constant exchange of ideas. Crick, with his loud laugh and even louder opinions, quickly became a central figure. His booming voice would often echo through the halls of the Cavendish. He was known for his sharp, analytical mind and his ability to quickly grasp the essence of a complex problem. He was not one for tedious laboratory work, preferring the realm of theoretical speculation. He excelled at synthesizing information from various sources to form a coherent picture. His official project was the structure of hemoglobin, but his mind was already on DNA. The pivotal moment came in 1951 with the arrival of a young American biologist named James Watson. Watson was brash, ambitious, and just 23 years old. He was singularly focused on discovering the structure of DNA. He believed it was the most important scientific problem of their time. He and Crick, despite their different backgrounds, found in each other a perfect intellectual partner. Crick was the seasoned physicist turned biologist, Watson the precocious biologist. They shared an office, room 103, in the Austin wing of the Cavendish. Their collaboration was characterized by an almost continuous conversation. It was a relentless brainstorming that pushed the boundaries of what was known. They were both convinced that the structure of DNA held the key to understanding heredity. They were in a frantic race to find it before anyone else. Their primary competitor was the brilliant chemist Linus Pauling in the United States. Pauling was a titan of science and was also known to be working on the problem. The fear of being scooped by Pauling was a powerful motivator. Duo's approach was unconventional for the time. They focused on building physical models from metal plates and wires. These models could incorporate all the known chemical and physical properties of DNA. They drew heavily on the work of others, a fact they readily admitted. A key source of information was the X-ray diffraction images of DNA. These were being produced by Rosalind Franklin and Maurice Wilkins at King's College London. Franklin, a meticulous and brilliant crystallographer, had refined the technique to produce incredibly clear images. Her images, especially the famous photograph 51, were crucial. This image, shown to Watson by Wilkins without Franklin's permission, was a revelation. It provided a vital clue, strongly suggesting that the DNA molecule was a helix. The distinct X pattern in the diffraction photo was the undeniable signature of a helical structure. It also allowed them to deduce key parameters of the helix. It was the piece of the puzzle they had been missing. Armed with this information, along with their knowledge of chemical bonding rules, Watson and Crick worked feverishly on their model. They built their large-scale model in the center of their shared office. They realized that the molecule was a double helix, not a single or triple one. The two strands twisted around each other like a spiral staircase. The sugar phosphate backbones formed the railings of the staircase. The nitrogenous bases were the steps, facing inwards. In a final flash of insight, they deduced the pairing of the nucleotide bases. Adenine, A, always paired with thymine, T, forming two hydrogen bonds. Guanine, G, always paired with cytosine. C, forming three hydrogen bonds. This specific pairing, known as complementary base pairing, was the secret. It explained how the two strands could be held together. It also ensured the diameter of the helix remained constant. This structure was the key to the molecule's function. It explained how DNA could carry vast amounts of information in its sequence of bases. Most importantly, it suggested a simple mechanism for replication. The two strands could unwind or unzip. Each strand could then serve as a template for the synthesis of a new, complementary strand. This elegant solution was a breakthrough of monumental proportions. In April 1953, they published their findings in a short, one-page paper in the journal Nature. The publication would become one of the most famous in the history of science. The paper modestly began with the sentence, "We wish to suggest a structure for the salt of deoxyribose nucleic acid, DNA, glycine." It ended with the now famous understatement, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." Lee? The discovery of the double helix was not the end of the story for Crick. It was merely the beginning of a new chapter. The structure answered the how of heredity, but it opened up an even more profound question. How does the sequence of bases in DNA direct the synthesis of proteins, the workhorses of the cell? This was the problem of the genetic code. Crick turned his formidable intellect to this new puzzle. He called it the coding problem. He theorized that the information in DNA was not used directly. It was transferred to an intermediary molecule, which he called messenger RNA. This molecule then carried the instructions to the protein-making machinery of the cell, the ribosomes. This led him to formulate what he called the central dogma of molecular biology in 1958. The central dogma stated that genetic information flows in one direction. It flows from DNA to RNA to protein. This concept provided a powerful and clarifying framework. It helped organize the thinking of the entire field of molecular biology. It was a bold and simple idea that has largely stood the test of time. Crick also proposed the adapter hypothesis. He suggested that an adapter molecule was needed to translate the language of nucleic acids into the language of proteins. This adapter molecule was later identified as transfer RNA, TRNA. Crick hypothesized that the code was read in groups of bases, not one by one. He reasoned that if there were only 4 bases, a code of single bases could only specify 4 different amino acids. This was far short of the 20 known to exist in proteins. A 2-base code could specify 16 4x4, which was still not enough. Therefore, the code had to be a triplet code. A sequence of 3 bases, a codon, would specify a single amino acid. This would provide 64 4x4v4 possible codons. This was more than enough to code for all 20 amino acids. In 1961, working with Sidney Brenner, Leslie Barnett, and Richard Watts Tobin, Crick conducted a series of elegant genetic experiments. They used bacteriophage T4 viruses to test their ideas. These experiments, based on inducing framshift mutations by adding or deleting single bases, provided the first strong evidence for a triplet code. They demonstrated that the genetic code was read in a specific reading frame. It was read from a fixed starting point, like words in a sentence. They showed that adding or deleting 1 or 2 bases would scramble the message downstream. This resulted in a non-functional protein. However, adding or deleting 3 bases would often restore the function. This was because it restored the original reading frame after the mutation. This experimental verification of the triplet nature of the code was a major triumph. It confirmed Crick's theoretical insights and paved the way for the complete deciphering of the code. While Crick and his team had established the general principles, the task of assigning specific codons to specific amino acids was taken up by others. Marshall Nirenberg and Hagobine Khorana led this effort. Using cell-free systems, they painstakingly worked out the meaning of each of the 64 codons. By 1966, the entire genetic code was cracked. It was discovered that the code was degenerate, meaning that some amino acids were specified by more than one codon. It was also found to be nearly universal, applying to all forms of life from bacteria to humans. This universality is a powerful testament to the common evolutionary origin of all life on Earth. For their monumental discovery of the DNA structure, Francis Crick, James Watson, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962.